Hi everyone, I'm Mariam Shemikh, a third year undergraduate biology student at York University. I'm an NSERC USRA student in the Rehan lab. The topic of our research is the epigenetic mechanisms of maternal care in Serotina calcarata. Serotina calcarata is the model organism used in the Rehan lab. So, what is Serotina calcarata? It's a type of carpenter bees. They are subsocial, commonly found throughout eastern North America from Ontario to Georgia. The Rehan lab has published the methylome, genome, and many transcriptome datasets for this species. They are known to prefer nesting in raspberry sticks and sumac trees as shown in the pictures. They are also known to be long-lived and care for their offspring throughout their development. Former studies in the Rehan lab showed that the social environment and absence of maternal care do affect gene regulation. And the picture on the left is that of a mother Serotina calcarata. And the picture on the right shows us the different stages of brood development in Serotina calcarata. Previous research regarding mechanisms of maternal care epigenetics showed that absence of maternal care can dramatically affect the gene expression in primates and rodents. For instance, it has been shown to increase the histone acetylation and cause methylation of certain regions of exons. In the offspring of Serotina calcarata, maternal care absence affects gene expression, DNA methylation, and alternative splicing of genes. Not only the gene regulation processes were proven to be affected by the absence of maternal care, but also the social behavior. Studies showed that the absence of maternal care in rodents, for instance, can increase aggression and anxiety. And in Serotina calcarata offspring, the absence of maternal care causes more aggression. Generally, absence of maternal care decreases the quality of early social behavior in juveniles. However, the timing at which maternal care is critical for gene regulation of offspring bees hasn't been studied yet, and the scope or extent of the effects of maternal care on epigenetics isn't clear yet either. So the purpose of our research is to determine which stages that maternal care absence can produce in pronounced effects on the behavior and gene expression in young adults of Serotina calcarata, and to determine the timing at which the absence will, in other words, severely affect the gene expression throughout different stages of the bee. Methods of this research include 4 steps. First step is to collect nests throughout the summer up until September, which is the active season of Serotina calcarata. Then we'll be performing behavioral assays or tests in order to observe the bees for 20 minutes and record all the interactions in the presence and absence of maternal care. Then we'll extract and isolate the RNA. At last, we'll perform statistical tests or analysis in order to detect the specific genes and regulatory networks affected by the absence of maternal care. So, why is our research important? And how can it be applied? It will help us understand more about the molecular mechanisms governing the developmental gene regulation, and it will help us understand more about the effects of maternal care on the brain and behavior of bees. That will definitely enrich our knowledge on social evolution, cooperation, and conflict in bees. At last, it will teach us more about the early childhood development, maternal care, stress, and anxiety response not only in insect societies but also in humans. At last, I'd like to thank all the Rehan Lab members, York University, and NSERC for their support of the research.